I would like to tell you a story about a young woman named Susan. Susan has been accepted into a top-tier medical school to pursue her lifelong dream of becoming a pediatrician. Over the next year, she noticed it became difficult for her to drive home after dark. She visited her eye doctor and was diagnosed with retinitis pigmentosa, or RP. Her doctor informed her, "Unfortunately, we have no treatment or cure to save your vision." Susan is one of 86,000 patients in the US that has heard this devastating news. RP starts out with a gradual loss of peripheral vision and will progress to complete blindness over several years. My lab studies RP because we want to help patients like Susan see their dreams. Within our eyes, we have two cell types that interpret light: rods, which are used for night vision, and cones, which are used for color vision. RP patients have rods that are sick and die, leading to the loss of night vision. Cones are not sick, but for reasons that aren't completely understood, they die, leading to the loss of color vision and eventually complete blindness. My lab believes that cones die due to oxidative stress. Oxidation is a chemical reaction similar to fire. Cells burn energy, creating oxidation and damage, but they can neutralize this using antioxidants, which are similar to fire retardants. My lab has been studying a protein called S1R that can activate the antioxidant defense machinery. I want to test whether activating S1R in a mouse model of RP can decrease oxidative stress to help maintain their vision. To test whether activating S1R can decrease oxidative stress, we utilize a dye that, if oxidized, glows. We found that RP mice with activated S1R had decreased oxidative stress and less cells were dying. This told us that activating S1R was able to protect the cells, but are they functioning? In order to test this, we can place an electrode on the eye and flash light. We found that RP mice with activated S1R had better cell function compared to those without. But this still left the question, is all this really helping our mice see better? In order to test this, we have an eye chart just for mice. We present a moving line, and if the mouse follows that line with its head, that confirms it can see. We found that RP mice with activated S1R could indeed see better. By activating S1R, we were able to prolong the vision of our mice. This groundbreaking evidence has the potential to prolong vision in patients who would otherwise have no treatment at all. My research brings us one step closer to finding a cure for RP and allowing patients like Susan to clearly see their dreams.